So the next talk will be from Xinhui Li, who is a graduate student at the Georgia Institute of Technology. She was also unable to come here due to visa issues. It seems the two graduate students both had those issues. And she's already said hi, so we'll go ahead and transition to her talk now. So.
Hi everyone, I'm Xinhui. I'm a fourth-year PhD student at Georgia Tech in Trent Center. Today I'll be presenting our work, Deep Generative Modeling for Latent Source Separation and Psychosis Continuum Estimation for Neuroimaging Data. Generative AI is revolutionizing how we create content, from writing poems to generating images. But how can we leverage these generative models to analyze neuroimaging data and gain insights into neurobiological mechanisms? In this talk, I will introduce two applications for neuroimaging analysis, one for blind source separation and one for estimating the psychosis continuum. Our blind source separation methods are developed within a unified framework called Multi-Dataset Independent Subspace Analysis, NISA. The original NISA framework includes three classic blind source separation methods, ICA, IVA, and ISA. Recently, we have expanded the capabilities of NISA and developed new methods to analyze multimodal neuroimaging data. I will introduce these new methods one by one. We first propose a method called multimodal independent vector analysis, MNIVA. We use the IVA module and NISA to jointly analyze multiple imaging modalities, including structural, functional, and diffusion MRI, and we identify the link sources for multimodal neuroimaging datasets. We find that several sources are significantly associated with phenotype measures, such as age, sex, and schizophrenia. From the brain spatial maps, we can see how these effects are expressed across three different imaging modalities. However, the limitation of MNIVA is that it assumes that the latent sources are one-dimensional and independent within each modality, but the underlying relationships among brain networks are likely more complex. For example, sources from the same modality might be grouped by their anatomical or functional properties. To relax the assumption in MNIVA, we recently developed a method called Multimodal Subspace Independent Vector Analysis, MSIVA. MSIVA aims to capture the linkage of vector sources by defining subspace structures that include both cross-modal and unimodal subspaces with different dimensions, as shown here. We evaluate three initialization workflows, each capturing different amounts of joint information. MSIVA first applies multimodal group PCA on both modalities, followed by ICA on each modality. We then perform combinatorial optimization and evaluate the MSIVA loss. This process is iterated until the loss converges. We apply MSIVA to jointly analyze both structural MRI and functional MRI in the UK Biobank dataset. Here we show the two MSIVA subspaces are significantly associated with age or sex. From the reconstructed brain spatial maps, we observe age effects in the cerebellum from SMRI and the occipital pole from fMRI. And we also observe sex effects in the frontal lobe and precuneus in both SMRI and fMRI. We further perform a Volsa-wise brain age delta analysis with MSIVA sources. This table here shows the number of Volsas with significant Pearson correlations between the brain age deltas and phenotypes. We find that the brain age gap can be explained by several phenotypes, like physical exercise, time spent watching TV, and sleep duration. Both MNIVA and MSIVA are linear methods. They assume that sources undergo a linear mixing process, but for neuroimaging data, it's likely that sources are nonlinearly mixed due to nonlinear transformations in many pre-processing and modeling steps, So these linear methods cannot effectively recover the nonlinear sources. To address this challenge, we propose a nonlinear method called Deep Independent Vector Analysis, DeepIVA, to learn nonlinearly mixed sources from multimodal data. DeepIVA is built upon two methods, Identifiable Variational Autoencoder, IVAE, and MESA. IVAE can recover nonlinear sources, and MESA can detect cross-model linkage. In DeepIVA, we use IVAE to identify unimodal sources, and then we use MESA to align cross-model sources. We iteratively update IVAE and MESA objective until convergence. We compare DeepIVA with several baseline methods. We have evaluated DeepIVA on synthetic data. Here we show the randomized dependence correlation matrix, which measures nonlinear dependence between two types of sources. In this figure, each column represents a method. The first two rows show the unimodal separation performance, one for each modality. The third row shows the cross-model alignment result, and the last row shows the ground truth cross-model correlation. If a method can identify unimodal sources, it should be able to see high values along the diagonal after column permutations in the first two rows. If a method can detect cross-model linkage, it should be able to see high values along the diagonal in the third row, just as the ground truth in the last row. We find that DeepIVA is the only method that can separate unimodal sources and detect cross-model linkage. We are currently working on applying DeepIVA on neuroimaging data to learn nonlinear neuroimaging sources. Next, I will talk about the functional network connectivity interpolation framework we developed to capture the neuropsychiatry continuum in heterogeneity. This framework aims to address some limitations of conventional diagnostic and analytic approaches. The current diagnostic system relies on subjective measures, such as expert assessment of patient self-report data, but these subjective measures are not enough to accurately capture biological deficits of mental disorders. Also, existing approaches often analyze group averages, but psychiatric disorders are highly heterogeneous and symptoms may exist along a continuum, so the group average approach alone is not enough. In addition, supervised classification approaches have been widely developed to predict disorder conditions. They require diagnosis labels as the input, but these labels are subjective measures, and they only indicate the current status of a disorder and cannot predict future disorder progression. Here, we use brain imaging data to learn objective, individualized biomarkers of schizophrenia. Specifically, we use the static functional network connectivity, SFNC, and dynamic functional network connectivity, DFNC, derived from the neural pipeline. We use the resting state fMRI dataset with schizophrenia patients and controls. We propose an interpolation framework based on an unsupervised generative model, a variational autoencoder, VAE. We train the VAE on SFNC and DFNC data separately. After we train the VAE model, we design 4 inference pathways. The first 2 pathways are for SFNC interpolation. To estimate continuous SFNC changes, we uniformly sample from the VAE latent space and visualize the generated SFNC matrices. To compare the generated SFNC and the original SFNC, we also feed the data to the trained encoder to extract latent features, and then we map these features to a 2-degree using a linear assignment algorithm, and then we can display the corresponding original SFNC matrices. The last 2 pathways are for DFNC interpolation. We apply a k-mean clustering algorithm on the VAE latent features, and we sample from the centroid of each cluster to get the generated DFNC states. We also perform k-mean clustering on DFNC data directly to derive the original DFNC states. Let's take a look at the results. Here, the figures on the left show the generated SFNC matrices while the figures on the right show the original SFNC matrices. Each small square is a subject's SFNC. We can see a high correspondence between the generative and original SFNC matrices. In the generative result, we observe a continuous change from the top half dominated by schizophrenia patients to the bottom half dominated by controls. And this 2-degree layout helps us to analyze group-specific patterns through pattern alterations, as well as individual variability. When we look at the DFNC states, we also observe a high correspondence between the generative and original states. For example, both generative and original states 1 are dominated by schizophrenia patients, and states 5 are dominated by controls. It suggests that our generative model can review group-specific dynamic states, and these group differences are generalizable on the test set. In the VAE latent space, we can generate SFNC along a trajectory of interest. Here, as we move from the control cluster to the patient cluster, we can visualize how the generated SFNC patterns change along the trajectory to estimate the psychosis continuum. Here are related papers in preprints. If you have any questions, please contact me by email. Thank you.